Hi, hello everyone. Welcome back to the session. So in today's class, we are going to discuss about the short answer question in the immune system. Okay. So this is in the second year. Okay. So in the series of explaining this long answer questions, short answer questions, and very short answer questions. So we have completed thoroughly the long answer questions regarding the first year as well as the second year. Okay. So we have completed the long answer questions. So now we are dealing with the short answer questions both in the first year as well as in the second year. Okay. So we are going to complete the short answer question. So after finishing the short answer questions, we are going to proceed with the very short answer question both in the first year as well as in the second year. Okay. So in today's class, we are going to deal with this immune system short answer questions in the second year. Okay, so the first question will be the writes a short note on the B cells. Okay, so just you should know the basic understanding of the blood cells. Okay, so in the blood cells we are having three different types of cells. Okay, RBC, WBC and the platelets. Okay, so this RBC is completely dealing with the oxygen carrying work. Okay, so this is the one carrying oxygen with the help of the hemoglobin pigment, and this WBC is the one involving in the blood coagulation, and this platelets is the one involving in the blood coagulation process. Okay, so with the help of the clotting factors. Now this WBC is the one plays a crucial role in the immunity. Okay, so in the WBC we are having five different types of cells. The first one will be the neutrophils, basophils, eosinophils, lymphocytes and monocytes. Okay, so these are all the five different types of WBC. So in the lymphocytes we are having two different types of cells, T cell lymphocytes and B cell lymphocytes. Okay, so this is the B cells we are going to deal with now. Okay, so write a short note on B cells. You should know that these B cells is coming under the category of lymphocytes. This lymphocytes is one of the five types of cells in coming under the WBC, and this WBC is dealing with the immunity. Okay, now what is the B cells? So lymphocytes produce the antibodies and capturing the circulating antigens. So you should know this lymphocytes. So as I have mentioned you before, so this WBC is the one plays a crucial role in the immunity process. Okay, so this WBC is having the five different types of cells: neutrophils, basophils, eosinophils, lymphocytes, and monocytes. So in this lymphocytes, this is the one. As I mentioned you before, this WBC plays a crucial role in immunity. Among all these five different types of cells of WBC, this lymphocytes is very important in the role of immunity. Okay. So this lymphocytes further divided into two different types of cells. One is T cell lymphocyte, another one is B cell lymphocytes. So in the B cell lymphocytes, further we are having two different types of cells. One is plasma cells and another one is memory cells. Okay, so this plasma cells is the one producing the antibody. Okay, so this plasma cells is the one producing the antibody. This memory cells is the one involving in the process of memorizing the process mechanism, how our immune system is killing the antigen when the antigen enter into the body for the first time. Whenever the antibody is entering, see, you have to understand the basic difference between this antibody and the antigen. So antigen is a foreign particles enter into our body. Antibody is the one against the antigen that is entering into our body. Our body is synthesizing the antibody to kill the antigen. So this is the basic understanding between this antigen and antibody. And this antibody is the one is secreted by the plasma cells, and the plasma cells is nothing but the type of the B cell lymphocyte. Now what is with respect to this memory cells? Whenever any antigen entering into our body will take some time to analyze the mechanism of the antigen, and they have to process the antigen and they have to produce the antibody. It will take some time. Okay, whenever the antigen enter into the body for the second time, this memory cells remember the process how we have killed the particular antigen when they entered into our body for the first time. Okay, so this memory cell is remembering the process. Okay, then the second time whenever the second time encountered by the antibody by the to the antigen, the process will be very faster, okay, when compared to the first time. So now you have to know that this is the lymphocytes, types of lymphocytes, that is the one producing the antibodies that capturing the circulating antigens. Now this B cells is the one originate in the bone marrow in the adults. So we are having T cell and B cell. Okay, so this T cell is going to produce in the bone marrow, and B cell is also produced in the bone marrow. Okay, so the T cells, they are matured in the thymus, and the B cells is matured in the bone marrow itself. So this is what the difference between T cells and the B cells. Okay, so as I have mentioned before, in adults the bone marrow is producing the B cells, and in the fetus the B cells will be produced in the liver, and in birds it will be produced by the bursa of Fabricius. So these are all the most important thing where the B cells is getting originating from. Now, where it is getting matured, the mature B cells displace the antibodies on their surface. See, whenever the antibodies are produced by the bone marrow, that is the B cells, whenever the B cells are produced in the bone marrow, it doesn't have any of the surface markers. Okay, doesn't have any of the antibodies on their surface. Whenever it is getting matured in the bone marrow, only after the B cells are getting matured in the bone marrow, they are having antibodies on their surface. And whenever the B cells having their antibodies on their surface, this cells is called as an immunocompetent B cells. So this is an important point. So three things you have to keep in mind. It is getting originating in the bone marrow, and it is getting matured in the bone marrow, and whenever it is matured, it is displaying their antibodies on their surface. As they are having the antibodies on their surface, matured B cell surface, it is called as an immunocompetent B cells. So what is mean by this immunocompetent B cells? In the sense, so this whenever the B cells are immunocompetent, it is ready to encounter the antigen. So that is what we are calling them as a immunocompetent B cells. Now what is its function? So this is the one migrate to the secondary lymphoid organs. So we are having lymphoid organs, okay, primary lymphoid organ and secondary lymphoid organ. What is meant by primary lymphoid organ? The primary lymphoid organ means thymus and bone marrow. So this thymus and bone marrow, we are calling them as the primary lymphoid organ. And in the secondary lymphoid organ, which includes spleen, okay, spleen and lymph nodes, lymphatic vessels, and other payers, patches in case of small intestine, all these organs are called as the secondary lymphoid organs. And also MALT, mucosa associated lymphoid tissue, gut associated lymphoid tissue, all these organs we are calling them as the secondary lymphoid organs. Once they have produced in the primary lymphoid organs, that is bone marrow, this B cells will be entered into the secondary lymphoid organs. Okay, so now after it has been migrated into the secondary lymphoid organs, they are differentiating into, see, first it is getting originated in the bone marrow. After it is getting originated, it is getting matured in the bone marrow itself. After they have matured, they will migrate into the secondary lymphoid organs. After they are once they are reaching the secondary lymphoid organs, they have to be differentiated. Okay, they have to differentiate. The B cells are now differentiated into plasma cells and the memory cells. Okay, now this plasma cells is the one responsible for producing the antibodies. Antibodies are nothing but an immunoglobulins. Okay, so we are having 5 different types of immunoglobulins, that is IgA, IgM, IgG, IgD, IgE. These are all the 5 different types of antibodies, and that antibodies are produced by the plasma cells, and this plasma cells is nothing but a differentiation of the B cells. Okay, now this memory cells. Now the B cells is not only differentiating into plasma cells, and it is also getting differentiating into memory cells. Now this memory cells is the one stores antigen information. So whenever the antigen enter into our body for the first time, it is analyzing the mechanism how our antibody is killing the antigens. Okay, they are storing the antigen information. So rapid response on the re-exposure. So whenever the antibody enter into our body for the first time, it will take some time to kill the antigen. Whenever the same antigen enter into our body for the second time, that is called as a re-exposure. This memory cells remember the process how we kill the antigen for the first time when it entering into our body. Okay, so this is what the function of the plasma cells as well as the memory cells. Now second question. So that's all about the first question with respect to the B cells. Now the second question will be write a short notes on the immunoglobulins. Okay, so this immunoglobulins, you should know what are all the, first you should know what will be their structure and what will be their function. Okay, so that is what that generally our antibody will be in the shape called as a Y shape. Okay, we can simply call them as in the Y-shaped antibody. Okay, so what are all the things present in the antibody? So antibodies are technically called as an immunoglobulin. Okay, so don't confuse with the word antibody and the immunoglobulins, both are same. And also you should know the difference between the two different words. One is epitope and another one is paratope. Okay, paratope is the one present in the antibody which is called as an antigen binding sites. Where the antigen is going to bind with the antibody, that site is called as an paratope. So paratope is the area which is present in the antibody which is called as an antigen binding site, where the antigen is going to bind with the antibody. Now epitope. Epitope is the area which is present in the antigen where the antibody is going to bind with the antigen. That is antibody binding site present in the antigen. We are calling them as an epitope. So this is the two important words you have to keep in mind. Now see, so this is a paratope. We are having a variable region and the constant region. Okay, so variable chain and the constant chain region. Okay, variable region. V stands for variable, C for constant, and this is called as a light chain and this is called as a heavy chain. So in the heavy chain there is only C region is present. In the light chain we are having V as well as C region, and this is called as an FCN, and this is called as an FAB ends. That is fragment of antigen binding site, fragment of the constant region, FC end. Okay, so these are all the important parts which is present in the structure of immunoglobulins. Now, how can we define the immunoglobulins? As I have mentioned you before, this immunoglobulins is also called as an antibody. Okay, so this antibody is produced by the plasma cells, and this plasma cells is nothing but, and this is the differentiation of the B cells. And the B cells are nothing but a type of the lymphocytes. Okay, so that is what in the lymphocytes we are having two different types of cell, T cell lymphocyte, B cell lymphocyte. So this lymphocyte, one of the type will be the B cell. Now the B cell is differentiating into the plasma cells, and the plasma cells is the one responsible for producing the antibody. Now this is the proteins. So immunoglobulins are generally proteins. Antibodies are nothing but a proteins. They are produced by the B cell lymphocytes. Okay, now the B cell lymphocytes is differentiating into the plasma cells. They are the one responsible for producing the antibodies. Now what is the point of having these antibodies, which is a protein? They are going to fight against the pathogen, and this pathogen is nothing but an antigens. Okay, that binds specifically to the antigens. Okay, and you should know the antigen and antibody, they are specific in nature. Specific antigen, specific antibody bind with a specific antigen. They are specific in nature. They are not general in nature. And paratope. So paratope, they are nothing but an antigen binding site of an antibody. Okay, so this is an antibody which is having the site that where the antigen is going to bind with the antibody. That site is technically called as a paratope. Now we are having a types by location. So based on the location where these antibodies are produced, we are having different types. Okay, so what are all the two different types of antibodies? We are having circulating antibodies, surface antibodies. Some of the antibodies are free in nature, they are circulating in the blood. Some of the antibodies are present in the cell surface. Okay, so that is what circulating antibodies, surface antibodies. So these antibodies are present in the body fluids, that is called as a circulating antibodies. Either these fluids may be, either it will be lymph or it will be the blood. Okay, and the surface antibodies, this is present in the B cells and the memory cells. Okay, so mature B cells, as I have mentioned you in the previous question, so this B cells, the mature B cells, definitely they are having an antibody in their cell surface. Only then we are calling them as a mature B cells. So those antibodies we are calling them as a surface antibodies. Not only in the B cells, it is also present in the memory cells. So only then the memory cells can remember the process. Now what will be the structure of the immunoglobulins? See, basically the structure of the immunoglobulin, it will be the Y shape. As I have mentioned you before, it will be in the Y shape. They are having 4 polypeptide chains. Okay, they are having 4 polypeptide chains, 2 heavy chains, 2 light chains. So that is why we can mention them as an H2L2, 2 heavy chain and 2 light chains. Okay, chains linked by the disulfide bonds. How these chains are linked together, that is called as a disulfide bonds. Okay, and we are having 2 ends, that is Fab ends and the FC end. So in the FC region, the cell is going to bind, and in the FAB region, the antigen is going to bind. Okay, so this is what an important structure of the immunoglobulins, and we are having 5 different types of antibodies: IgD, IgE, IgG, IgA and IgM. Okay, IgA is the antibody, it is present in the secretions. Okay, secretions in the sense it is present in the saliva region, or it may be in the tears. Okay, so the colostrum, that is the first milk given by the mother to the child, so that is colostrum. So all in the secretions we can find the IgA antibody, and in the IgG is the antibody, is the only antibody which has the capacity to cross the placenta. This placenta is nothing but this is the layer which is covering, which is connecting the baby to the mother. Okay, and this is the only antibody which can cross the placenta. And IgM is the region whenever if there any antigen is encountered by our immune system, this is the first antibody coming to the spot. Okay, followed by the IgG. Okay, so now IgE is the one responsible for the allergy reaction. Whenever there is a particular person is experiencing with an allergy, they are having this IgE antibodies in higher proportion. Okay, IgD, they are involving in the antigen presentation. Okay, antigen presenting antibody. Okay, so this is what that's all about the second question, that is a write a short notes on immunoglobulins. Now coming to the third question, describe the various types of barriers of innate immunity. So basically in our immune system, this is an immunity, we can classify this immunity into 2 types. Okay, so this immunity can be classified into 2 types. One is innate immunity and another one is adaptive immunity. Okay, so innate immunity in the sense those immunity we are having by birth. Okay, adaptive immunity we are getting acquired from the after we are getting born. Okay, so this is what a basic difference between innate immunity and adaptive immunity. And the innate immunity are non-specific in nature. This adaptive immunity will be specific in nature. So these two are the major difference between this innate and adaptive immunity. Now this questions is dealing with the barriers of innate immunity. Okay, so how can we define this innate immunity? So first it will be inborn. Okay, these are all the immunity we are having by birth itself. Okay, now this is non-specific resistance to diseases. Whatever be the diseases entering into our body, this is the first line of defense. Okay, so this is non-specific in nature. Either it will be bacteria or virus or fungi, whatever be the microorganisms will be, this is non-specific. I am ready to face you. So this is what non-specific resistance. This acts without a prior exposure to the pathogens. Okay, so as I have mentioned you before, we are having memory cells. So this memory cells is the one remembering the process whenever the antigen enter into the first time, how our antibody kill the antigen. Okay, so this memory cells remember the mechanism. Now this innate immunity doesn't have any memory cells. They act without any previous exposure. Okay, so this is what non-specific, they act without any previous exposure, and this is inborn. Okay, now what are all the barriers? Generally they are having 4 barriers in case of innate immunity. And the first barrier will be this physical barrier, physiological barrier, cellular barrier, and cytokine barrier. So these are all the 4 different types of barrier present in the innate immunity. Now in the physical barrier, we are having skin and the mucous membrane. So in the skin, so they are preventing the pathogen entry. Okay, skin is the one preventing the pathogen entry. How the skin is preventing the pathogen entry? So 2 things you have to keep in mind with respect to the skin. So the largest organ in our body, that will be the skin. Okay, no doubt in that. Our entire body is covered by a organ which is called as a skin. Okay, and the second important thing will be the pH maintained in the skin. Okay, so in the summer time we used to sweat. Okay, and that sweat, they are having an acidic environment, though that pH will not be the suitable environment for many of the microorganisms. So the skin plays a crucial role in preventing the microorganisms. So this is how the skin is involving in preventing the pathogen entry. Now, the second thing coming under the physical barrier will be the mucous membranes. Okay, so this mucous membrane is the one. So the most important thing, the 2 important thing you have to keep in mind with respect to this mucous membrane will be the first thing. The mucous membrane all over body, our entire gastrointestinal tract will be covered by this mucous membrane. Okay, and the second thing will be the texture. The texture of this mucous membrane, that is the texture of this mucus will be sticky. Okay, once the microorganism will be touch the mucous membrane will going to trap the microorganism so that it can't escape. So this is what the 2 important things. So the mucous membrane trap the microorganism which is entering into other body. So the skin and the mucous membrane which is coming under the physical barrier. The second important barrier will be the physiological barrier. Okay, the physiological barrier in the sense, in our stomach we are having an HCl. See, this HCl is nothing but an hydrochloric acid. Okay, so this is a hydrochloric acid. As it is an acid, again the pH will not be suitable for most of the microorganisms, so that it will also be against the microorganisms. Also in saliva and tears. So in the saliva and in the tears, we are having an important enzyme that is called as a lysozyme. So it is not a lysosome. Lysosome is a cellular organelle. We are calling them as a suicidal bag. That is different. Lysozyme is an enzyme which has some antimicrobial activity that is present in the secretions like saliva and tears. They are the one involving in destroying the microbes. Right? Now, cellular barriers. Now these are all the important barriers. So the cellular barriers, we are having an immune cells. That cells is nothing but a WBC. Okay, so in the WBC we are having a neutrophils and in the monocytes, and also we are having an NK cells, that is natural killer cells, and macrophages. Based on the origin of this natural killer cells, we didn't have a proper information who is the progenitor for this natural killer cells. Okay, now this macrophages. So all these are the immune cells. They are the one involving in the process of phagocytosis process. Phagocytosis in the sense, they are the one involving in engulfing the microorganism and digesting. Okay, that process is called as a phagocytosis. So these are all the cells coming under the cellular barriers. Now comes the cytokine barriers. Cytokines are secreted by the immune cells. Okay, cytokines, they are secreted by the immune cells. So this is the one secreting by the immune cells. What is the role of the cytokines? They are helping the immune cell immune cell differentiation and protecting the uninfected cells. Okay, so if one cell is getting infected, obviously the other cell is also going to infected by the microorganism. So the uninfected cells are getting protected by the cytokines, and the cytokines are helpful in the differentiation of the immune cell. So these are all the 4 important types of barriers of this innate immunity. Okay, next comes the humoral immunity. See, I will explain you. So as I have mentioned you before, in the WBC we are having 5 different types of cells. Okay, so in the 5 different types of cells, the most important cell will be the lymphocytes. Okay, so in the lymphocytes we are having 2 different types of cells, that is B cell lymphocyte and T cell lymphocyte. Okay, so we have studied about this 4 different barriers which is coming under the innate immunity. So we are having 2 different types of innate immunity, innate immunity, and another one will be the adaptive immunity. So in innate immunity we have studied the 4 different types of barriers. In adaptive immunity, so this lymphocytes, we are calling them as the adaptive immunity. So in the adaptive immunity, we are having 2 different types of cells. One is B cell lymphocyte and another one is T cell lymphocyte. If the T cell lymphocytes are involving in the immunological process, we can call them as the cell mediated immunity. If the B cells are involving in the immunological process, we are calling them as the humoral immunity. Okay, the word humoral means fluid. Okay, and the fluid, these fluids, they are having an antibodies. The antibodies are produced by the plasma cells. Again, plasma cells is the differentiation of this B cells. Okay, so this is the basic understanding of this humoral immunity, adaptive immunity, and innate immunity. Now comes the humoral immunity. Okay, so now in the humoral immunity, so what now? Both T cells and B cells are going to involve. What is the function of the T cell and what is the function of the B cells? Ultimately, antibodies are produced by the B cell. Okay, but before that, before the B cells are producing the antibody, the T cell has to present the antigen to the B cell. How it is happening? Okay, so see here, this is what T helper cell. See, in the T cells, we are having 3 different types of cells. One is suppressor T cell, cytotoxic T lymphocyte, and the helper T cell. So this helper T cell, we can denote them into TH cell. Okay, so in the helper T cell, so this is the cell. Okay, so each cell having the receptor. Okay, so like in the same way, the T helper cells, they are also having the receptor. So that is called as a T cell receptor, TCR. Okay, so not only the receptor, they are also having the surface marker, and that surface marker is nothing but CD4. Okay, T cell receptor, TCR. CD4 is a surface marker that is present in the T helper cell, that is nothing but a CD4. Okay, now see here, this is a mature B cell. So this is a B cell. So in the surface of the B cell, we are having an antibody, that is Y-shaped antibody. Okay, so this is what a B cell. Okay, now see, this is what a T helper cell. Okay, now this is the receptor which is present in the T cell is called as a T cell receptor. And the T cell receptor, besides the TCR, they are also having the CDR marker which is also present in the T helper cell. Okay, now because of this TCR, CD4, this T helper cell is identifying the antigen, helping the B cell to identify the antigen. I'm sorry. Okay, so this is what a B cell. Now they are producing an interleukins. Okay, so this interleukins, now this B cell, they are differentiating into the plasma cell and the memory cell. So the plasma cell is the one releasing the antibodies, and the memory cell is the one remembering which antibody is suitable for killing the antigen whenever the antigen entering into the first step. So this is what an interaction between the T cell as well as the B cell. Okay, now antigen recognition. How it is going to identify the antigen. So how the antigen recognition. So as I have mentioned you before, this humoral immunity, in the sense B cells are involving. Okay, B cells. Okay, if T cells involved, that process is called as cell mediated immunity. Okay, so as it is a B cell, it is the B cell humoral immunity. Now they are the one has to recognize the antigen. Free antigens binds to the Fab region. Okay, so this is the free antigens. The free antigen which is circulating in the blood, they are the one binding to the Fab region. Fab region is the one which is present in the antibodies. Okay, that is what fragment of antigen binding region. Okay, so this antigens which is present in the mature B cells. Okay, the mature B cells contains the antibodies. In that antibody we are having a Fab region. So in the Fab region the antigen going to bind with that region. Now, the B cells engulf and process the antigens, displace the fragments via the MHC complex molecules. MHC in the sense major histocompatibility complex. Okay, so this is the cell that plays an important role in differentiating the self and non-self cells. Whether this is a non-self or this is a self region. Self cells, okay, non-self in the sense antigen. Self cells is a cell which is present in our body. So that differentiation will be done by this major histocompatibility complex. Okay, so this is the important thing. Now, once the antigen is getting recognized, the T cell is going to activate. Okay, T cell activation, that is specifically the T helper cells. T helper cells recognize the antigen which is present identified by this MHC complex. See, MHC complex is the one identify whether this will be the self or non-self. Okay, if the MHC complex identified that particular cells as a non-self cells, the T cell is going to activate. That once the T cell is getting activated, specifically the T helper cells, then it is going to recognize the antigen. Now, the T helper cells, they are the one involving in releasing the interleukins. Okay, so once they are releasing the interleukins, this interleukins is the signal for the B cells. This interleukins is the one stimulating the B cells. Now the stimulate is now the stimulation of these B cells, they have got the signal. They have to proliferate and they have to differentiate into plasma cells and memory cells. This is what a process. Okay, so this is what the mechanism of the humoral immunity. Next question will be this mechanism of the cell mediated immunity. Okay, so this is what an important thing. So cell mediated immunity, in the sense which cell is going to involve here, T cell is going to involve in the immunological process. How? As I have mentioned before, so this is the T helper cell. Okay, so this is the T helper cell. The T helper cells, they are having a surface marker which is called as a CD4 cells. Okay, so see here, there is also a major histocompatibility complex, that is MHC molecules, and this APC is nothing but an antigen presenting cells. Okay, antigen presenting cells are nothing but a macrophages and dendritic cells. So these are the cells which we are calling them as the antigen presenting cells. So once the cells, so this is an antigen, they are make them into fragments. Now these fragments, they have been identified by this MHC molecules as non-self cells. The T helper cells is going to activate. Now this T cells, they are going to release the interleukins. Once the interleukins has been released, this is not only activating the B cells, but it is also activating another T cell that is called as a TC cell, that is cytotoxic T lymphocyte. See, as I have mentioned before, T cell lymphocyte, further they are having three different types of cells: cytotoxic T lymphocyte, helper T cells and suppressor T cells. So these are all the three important cells, types of the T cell. Now T helper cells is the one involving their role in release of this interleukins. Now once the interleukins has been released, the TC cell is going to activate, that is cytotoxic T lymphocyte. So CD4 is the marker which is present in the surface of the helper T cell. CD8 is a marker which is present in the cytotoxic T lymphocyte. Okay. Also this TC cell is also having the T cell receptor. Okay, and another important thing will be here the MHC complex 2 will be present in the T helper cell, and MHC complex 1 is present in case of TC cell. Okay, now once the TC cell, This TC cell is also having a memory T cell and effector cytotoxic T lymphocyte, that is TC cell. Okay, this TC cell is also differentiating into two different types of cells, just like B cell, that is memory T cell, and the effector CTL, which means cytotoxic T lymphocyte. Okay, now this cytotoxic T lymphocytes, they have been developed into a perforance, okay, or pores. So this is the infected cell, which a cell is going to infect, and they are also, because of this granzymes and the process of apoptosis, the infected cells is going to digested by the cell mediated immunity. Let me explain once again, little more deeper. Okay, see, first process will be antigen processing. How the antigen is getting processed? Exogenous antigen, endogenous altered self-antigens. We are having two different types of antigen. What is the difference between these two? Exogenous antigen, in the sense, they are the antigen processed by the antigen presenting cells. Okay, so those antigen which are processed by this antigen presenting cells, we are called them as the exogenous antigens. Endogenous or altered self antigens means they are processed by the infected or altered cells, which means that cell, which is cells that is getting infected by the antigens. So those cells are called as an endogenous. Now, the antigen fragments displayed on the antigen presenting cells altered cell membranes. Okay, now these are all the antigens. So these are all the fragmented antigens. So these antigens, they are displayed on the antigen presenting cells. Okay, once the antigen presenting cells displaying the antigen, the T cell is going to activate. Okay, so now in the T cells, they are having an MHC complex involving in differentiating self cell and non-self cell. They are the one form the activated T cells and the memory T cells. Now the activated T helper cells secreting the interleukins. Once the interleukins has been secreted by the helper T cells, they are stimulating the T cell lymphocyte, that is cytotoxic T cells, to become the cytotoxic T lymphocyte, CTLs. Now, once the CTL has been produced, the infected cells, they are going to destructed by the CTLs. Okay, by releasing the perforins and the granzymes. Once the perforins mean they are going to form a pores in the infected cells, and the granzymes is the one, they are the enzymes inducing the process of apoptosis. Apoptosis is nothing but a programmed cell death. Okay, now what is the point of having this NK cells? That is natural killer cells. Natural killer cells functions like a cytotoxic T lymphocytes but act as antibody independent. Okay, whereas CTLs are antibody dependent. Okay, T cells. We are having T helper cells, TC cells. Okay, this T helper cell is going to activate the B cell, and the B cell is going to produce the antibody. So this scenario is called as an antibody dependent. Okay, now this NK cells is also can function as a cytotoxic T lymphocytes, but it is not going to stimulate the B cell, but instead they themselves has the capacity to kill the infected cells. So that is why we are calling this NK cells as the antibody independent. The CTL cells are antibody dependent, because this T cell is going to stimulate the B cells, and the B cells going to differentiate into plasma cells, and the plasma cells going to produce an antibody. As they are antibody dependent, we are calling them as the CTLs or antibody dependent. Now another important question will be the AIDS. Explain the mechanism by which the HIV multiplies and leads to AIDS. AIDS is nothing but acquired immunodeficiency syndrome, and this HIV is human immunodeficiency virus. Okay, so this is how the virus will be. So the outer layer is called as in GP120. GP120 in the sense glycoprotein 120. Okay, and this layer is called as a lipid layer. So this layer is called as a lipid layer, and the genetic material here will be the RNA, no doubt in that is an RNA. And in the RNA, there is an important enzyme present in the RNA which is called as a reverse transcriptase enzyme. Okay, and the genetic material which is covered by a protein coat. Okay, and there is a layer of proteins, and here this is called as in GP41. This GP41 we are calling them as a glycoprotein 41. Now, how the HIV enter into the human body? The HIV, human immunodeficiency virus, enters into the T helper cells, macrophages or dendritic cells via the CD4 receptors. So we have studied before, the CD4 marker is very important for the T helper cell. So then only they can identify whether this is an antigen or this is R cells with the help of the major histocompatibility complex. So if the CD4 marker will be destroyed by this HIV, the T helper cells is functionless. The macrophages and dendritic cells, they can't act as an antigen presenting cells because of the absence of the CD4 cells. Okay, so this HIV, that is GP120, that is glycoprotein 120, that is bind with the CD4 marker, that is the CD4 receptor. Now the reverse transcription process will be taking place. Reverse transcription in the sense, see, initially the DNA has to be converted into an RNA, and RNA will be converted into protein. Okay, DNA to DNA is called as an replication. DNA to RNA is called as an transcription, and RNA to protein is called as a translation. Okay, so this DNA will be converting into an RNA is called as a transcription. RNA converting into a DNA is called as a reverse transcription, and this reverse transcription will be mediated by an important enzyme that is called as a reverse transcriptase enzyme, which is already present in the virus, HIV virus. Okay. Now, see, RNA will be converting into a DNA. So, this is what a reverse transcription process mediated by the reverse transcriptase enzyme. In the human body, we are having a DNA. This HIV is having the RNA. Now, with the help of this reverse transcriptase enzyme, the reverse transcription process is going to take place. Because why it has to perform this reverse transcription? Because the host, that is human, they are having a DNA. So that the virus is transferring their genetic material RNA into DNA, so that our host, our human immune system, as the CD4 markers have been destroyed, both are the DNA, they didn't identify whether this particular HIV is an antigen. So this is what the scenario. Our entire immune system has been compromised here. Now, this HIV single-stranded RNA, complementary DNA, they have been converted into the complementary DNA with the help of the reverse transcriptase enzyme, and this process is called as a reverse transcription process. Now, the second DNA strands forms. As the complementary DNA forms, this single-stranded RNA has been converted into the double-stranded DNA. Double-stranded, it is a viral DNA. Now, this viral DNA that will be integrated into the host DNA, that will be integrated into our host DNA, that with the help of an enzyme called as an integrase enzyme. Now the host cell, now what will be our host cell will do? Our DNA, that is a viral DNA, our host enzyme that will involve in the replication process, transcription process, translation process, and in the translation process protein will be synthesized, and the synthesized protein here, it will not the human DNA, that will be the viral DNA. This is how the virus is going to produced. Now the viral components will be assembled, new HIV particles bud off, and together they are going to assemble as a new virus. What will be the effect of our immune system? Continuous HIV production, progressive loss of the T helper cells. As we are continuously losing our HIV, losing our helper cells, our immune system will be totally compromised. So that is why we are calling them as an immunodeficiency. So this is why we are calling them as the human immunodeficiency virus. Okay, so that results in the syndrome which is called as an acquired immunodeficiency syndrome. So acquired immunodeficiency syndrome, it is not a disease, it is a syndrome, which means collection of many symptoms. We are calling them as a syndrome. Once our immune system has been compromised, many bacteria, many virus, many fungi can be easily affect the human body. So that is why we are calling them as an acquired immunodeficiency syndrome. Tissue trophism. What is tissue trophism? Here HIV is specifically attacking the T helper cells. The marker which is present is called as a CD4 markers. Okay, sparing the macrophages. So this is what the important mechanism of the HIV which is doing in our human body. Okay, so that's all about this immune system. Okay, hope the session will be useful for you. Thank you so much. We'll meet you in the next short answer questions chapter. Thank you.